Only those who dare to fail greatly can ever achieve greatly. Robert F. Kennedy. Hello Shishas, welcome to lecture 5 on cell division. We are going to we already seen about the importance of cell division. So the cells are the unique structures that you find inside the living organism, and they play a major role in transferring the genetic material and also the various functionalities. That's why you can see all the living organisms of their own species, all the cells work in a specific way. Based on the way the cells divide in the all living organisms, we have classified them into three major groups. One is amitosis, mitosis, and meiosis. If you look at the importance of cell division, it maintains the cell size. If you can remember properly, the surface area of cell is important. Whenever the cell surface area is being increased, so it leads to the less productivity of cell. Hence, they go for the cell division. And then the repair of cells. The cells are often has to repair themselves whenever there is a damage happened. And next is reproduction in organisms. It's quite evident that in basic organisms or the unicellular organisms, they show mostly this specific type of division, and they're going to divide themselves. Okay, it doesn't have any other use. Even cell division itself has a ability to divide. Okay, and then we have to know a few things about the cell division. If you look at last time, we discussed about the cell cycle. The cell cycle has two stages. One is the division stage, and the one is how the cell is going to maintain its own growth. Okay, it doesn't make sense when cell is not caring about its growth. It's only focusing on division, means it is not at all going to be a healthy cell, isn't it? That's why the cell always demands the proper growth, and then it's going to opt for the division at the particular time. The cell division will be carried out in two steps. One is the karyokinesis, the nuclear division. Remember, the prefix karyo refers to the nucleus. And next you can see the cyto. The prefix cyto refers to the cell, or even sometimes cytoplasm. And kinesis means division, so the cytoplasmic division. Amitosis, there was a brief introduction last time. This event takes place in two steps. The nuclear division takes place without a definite change in the chromosome, and second is the cytoplasmic division. So here the direct division. So that's why even the amitosis is also called as the direct division, because of the splitting of nucleus and then the centripetal construction to divide two daughter individuals. Examples for amitosis, or where we see this type of division, is Paramecium euglena, and even budding in uh, yeast is also considered as one of the example for amitosis. Let's look at the mitosis. Mitosis is also called as the equatorial division. We look at wherever the mitosis is going to occur, it's going to revise the exact copy. It's like this, whenever you put some paper in Xerox, when you want to go for a replicated form, it's going to produce an exact copy. In the same manner, the mitosis is also going to do such things. It's going to produce similar set of the things whenever they come out of the other side. Before going into the cell division, I wanted to show this graphical form of the cell cycle because it makes quite sense. The first step you can see here is the interphase. The interphase is where the cell is going to grow definitely, and this Z0 phase we can say, so it's also called as the interphase. So after interphase you can see the G1 phase, or the gap 1 phase. This is the first phase of growth, and it develops all its organelles, and it be ready for the synthesis of DNA. So that's why when this is called as the pre-DNA, pre-S phase. And here you can see the term itself says S refers to synthesis of DNA, or S phase, and G2 refers to the gap 2. As soon as DNA got replicated, cell doesn't prefer to go for the division, but still it waits for some time. It makes sure that all other organelles are ready to perform its activities. So that's why the gap 2 is followed before the cell goes for the M phase. Try to understand the M might refer to mitosis or meiosis. Mostly meiosis may have only one division and mitosis will have many divisions. So under mitosis you can see again the first cytoplasm, it's only talks about the karyokinetic phases. It doesn't have any clue about the cytokinetic division. First one is prophase, another one is metaphase, and then anaphase, telophase, and next we can see second set of reactions called cytokinesis. So these are all the mitotic phases, meiotic phases will be arranged. Alright, so it's about cell cycle. Now let us go back and understand more about mitosis. So as you know, the mitotic phases carries out in two different steps. One is called as the karyokinesis, another one is called as cytokinesis. We give more emphasis on karyokinesis. The reason is the nucleus is one of the important structure, and the things that actually divide is called as the chromatin or chromosomes. So the karyokinesis or nuclear division will have four different phases. One is called as prophase, second one is called as metaphase, third one is called as anaphase, and fourth one is called as telophase. So PMAT. And here we look at the prophase. The term itself says pro, that means beginning. So it is one of the longest phase. The reason is the various structures in the nucleus has to prepare for the cell division. The first thing you can see here is the chromatin condensation. If you remember correctly, I told you the chromosomes will only appear during division. So the chromatin threads, they are always in rest, like small threads, they will be in rest condition. Whenever cell wants to divide, the chromatins will come very close. They tighten up like this. So that's why you can see the chromosomal structure or dumbbell structure. And next you can see some of the important obstacles for cell division will disintegrate. The reason of disappearance of this is to facilitate more area for the moving of the spindles, you can say. First is nuclear membrane will disintegrate, and then you can see nucleolus starts disappearing. It doesn't make any sense when there is nucleolus inside the nucleus when there is no need of any proteins production. That's why nucleolus start disappears, and then you can see the centrioles that separates and start move towards the opposite poles of the cell. So this is a major event. You can see three important steps. First step is chromatin condensation. Second step is where the nuclear membrane disintegrates and nucleolus start disappearing. And thirdly, the centriole gets duplicated and they move towards opposite poles of the cell. Based on the shape they form for the attachment of the centromeres, we can divide the cell divisions as ancestral mitosis and ancestral mitosis. The ancestral mitosis generally seen in case of the plants because the centrioles are absent in them. Instead of the centrioles, the plants will have something called the polar bodies. The polar bodies are going to carry out the function of cell division. And next is called as ancestral mitosis. The ancestral mitosis refers to the formation of esters or the star-shaped structures while they are attached at the centromere region here. So that's why it is called as the ancestral. It's clear this is one of the major differences between the plant cell and animal cell when you observe in a macroscopic way. Clear? Let's move to the next step called as metaphase. The metaphase is the second step. It is helpful. Whatever the connectivities are produced, these are going to move further. In metaphase, what happens is the major changes is the chromosomes are arranged at the equatorial plane. All the chromosomes will come and they lay at the central portion of the cell, and then the centrosome will be joined. So this is the exact time where the centrosomes will be attached by spindles. So spindles are nothing but small thread-like structures. So just imagine the chromosome is here, and then when the chromosomes were duplicated, you can see two sets of chromosome. So that's why in this condition we can call the chromosome in the organism as foreign. The reason is there are two sets of chromosomes, and the spindle fibers will be produced by centrioles, and they attach at a particular region. So this region, wherever the attachment is taking place, it is called as the region of kinetochore. So you can see the kinetochore will be here, and once it gets attached to the kinetochore, and all of them are going to be pulled apart. And here you can see the replicated chromatids will be clearly visible. So this is a stage. Even we are going to look at the live images as well. You can understand more about them. And next, thirdly, is anaphase. Next we have seen that the centromeres have been pulled or attached by the spindle fibers. The next step, what happens is, as I told you, whenever there is 4n, it should supposed to be divided equally. That means one part of cell has to get 2n, and the part has to get 2n. For this, what happens is the centromere, exactly this region, is going to get duplicated, and it's going to be given to two parts of cells. So like this, this is centromere, and just imagine like this, the centromere is going to get duplicated. Remember this point very clearly. The centromere duplication is a major change, a major cause for the equal distribution of the chromosome. Alright, so one side goes one side, and this side it goes the another set. So that's how it maintains a similar set of chromosomes. Clear? Next is anaphase. In the anaphase, you can predict, these the duplicated ones are going to move towards opposite directions. This will be monitored by the spindle fibers, the thread-like structures that are joined at the kinetochores. The kinetochores are going to, they just shrink. You can say they will be start pulling up towards the centrioles in the opposite directions, and this makes sure that they are moving towards the polar regions of the cell. That means the end of the cells. And even this will be, this is a stage where you can see the cytokinesis may start at the end. So that's why it is called as this is even called as the stage where it happens is called as late anaphase as well. All right. Even though we see cell division as a simple thing to understand, but still there will be a lot of research by cancer scientists. The people who study about cancer, they focus a lot about all these structures, because everything is very essential for the growth. When there is overgrowth, there could be some problem here to continue it. Okay, let us continue with the last stage, the telophase. The telophase is exactly called as reverse prophase. The reason why we call reverse of prophase, because as you see in prophase, the centromeres the nucleolus will be disappearing. So here the nucleolus is going to reappear, and then you can see some other changes, like it's going to get the nuclear membrane back. So that's why it is called as the reverse prophase. And then I missed one more, where you can see the spindle fibers will be disappearing, and then centrioles will be getting joined with the cytoskeleton of the cell. So that's how the telophase is considered as the reverse of prophase. And next important division we're going to look, the important stage of cell division, is the cytokinesis. The cytokinesis is where the cytoplasm division takes place. The cytoplasmic division is quite obvious, because it is not a solid structure, it is like semi-solid structure. So obviously there should be some division happening with the help of the proteins that are produced before the cell division. Okay, here you can see it happens in two ways, particularly in plants one way, in animals in a different way. So when you look at the animal cytoplasmic division, it goes with a method called constriction and furrow formation. So the constriction is going to divide the organism into two major parts, and then you can see two daughter individuals are formed. But the similar method cannot be applicable for the plants. The reason is we have a strongest boundary called cell wall. So this rigid cell wall won't allow the plant to go for constriction or for a formation. So that's why plants are going to produce the cell plate. The cell plate is nothing but a chain of proteins. The chain of proteins are going to stay at the middle, and both on the other both sides you can see the new cells are being produced. So this way the cytoplasmic division in plants will be done by the cell plate method, okay, or even protein plate method. So these are the major differences. And when you look at the significance of mitosis, it is almost the repeated part. You can see in the beginning the growth and development, the genetic stability, repairing, healing, and regeneration. Almost every part of your body is produced because of mitosis. And remember, if there is 2n condition of cell, when it undergoes the mitotic division, it's going to produce the exact copy of the chromosomes. It doesn't go for producing any other set of chromosomes. So this is a major significance of the mitosis. Now let us look at some of the visuals before I close the lecture. I end the lecture here. Maybe some books even claim that early prophase, late prophase, early telophase. So we can divide the stages into more even. So first let us look at the early prophase. You can see the chromatids at the rest. The chromatid threads are rest, and then they have strong nuclear membrane. Maybe nucleolus is not shown here, but it will be there. And then you can see gradually it just disappears, and then you can see the nuclear membrane is getting disintegrated, and then chromosomal condensation is being formed. So in this stage probably you can see the structure of chromosome partially, and then you can see the centrioles moving up. I think this part is missing. It's quite difficult to find the scientifically good picture, the diagram. So next we can see the centrioles are being producing the spindle fibers, the thread-like structures here. So these are attaching at the particular region called as the kinetochores. After that you can see the metaphase, exact metaphase. You can see all of them are being present in the middle of cell. So middle of cell, and then next we can see in the anaphase the chromatin fibers are being condensed. They are pulling towards the poles, and then at the late anaphase you can predict. So slowly the constriction is being produced in case of animals. The cytokinetic division may also start. And then in telophase you can see the nuclear membrane is being produced, and then gradually the chromatin threads are going to pack up like threads. And then finally you can see the two newly produced cells, but exact DNA copies. Makes sense? And I found one of the teachers was trying to explain in a beautiful manner, so even I want to tell you how she did. So I think in class I told you. So P refers to prepare. So during the division the cells are going to prepare in the prophase, and M refers to the middle. So the cells are going to stay at the middle during this phase. And next A refers to apart. You can see in the anaphase the chromosomes are being apart. And next telophase T refers to 2. Nice, isn't it? Let's look at some of the models here. So here you can see it's the exact way how it happens inside the plants. So I got this from an atlas. You can see the chromatin condensation here, not exactly, but still somehow you can see the centromeres are being grown up, okay, bit by bit. And in the next stage you can see the nuclear envelope is getting disintegrated. So slowly you can see how the disintegration is happening, and nucleus is almost absent. May not see that clearly here. And next is a metaphase. All of them are middle of the cell, the mid plate of the cell. So even it is called as equatorial plate. So equatorial plane of the cell. You can see the centromeres attachment here. And next we can see the late metaphase. So the late metaphase, you can see exact condensation, and then the spindle fibers are attached there still. And then you can see the early anaphase, so gradual movement. So the separation is happening slowly, so that they're moving away from the center plate. So now you can see, like, okay, they just moved to opposite directions completely. Okay, so you can see almost the ends of the other ends of this cell as well. Next, after anaphase, we have two stages. Okay, the telophase and then the interphase. In the telophase, you can see the cell plate, because this is an example of plant hyacinth, the water hyacinth, which is about 430x resolution. You can see clearly how these daughter chromosomes are found. Okay, the cell plate is produced, and then the cells are separated. Again, the condensation is happening, and you may see the nucleolus, nuclear membrane production. And this is interphase. Like, you don't see nucleus much, and the chromatin fibers are submerged. And finally, before saying goodbye to you, I wanted to show an animation. We can understand through this more. Here is okay, ready, set. This side you can see them, it's in metaphase right now. Centromere division moving towards opposite poles, and then there is a cell plate that is being produced. Then the telophase, the new cells are formed, and they are decondensing, and condensation is taking place. Nucleolus, nuclear membrane. So that's it from me, Shisha. So